Hey there, scientists! Today, I'm diving into the fascinating world of immunoprecipitation. It's a powerful technique for isolating antigens by binding them to specific antibodies attached to a sedimentable matrix. But let's break it down a bit more. First off, where do we get our antigens? They can come from a variety of sources: unlabeled cells or tissues, metabolically or extrinsically labeled cells, subcellular fractions, or even in vitro translated proteins. And it's not just about isolation. Immunoprecipitation is also great for analyzing protein fractions, separated by other biochemical techniques like gel filtration or density gradient sedimentation. Now, let's talk antibodies. Both monoclonal and polyclonal antibodies from various species can be used. These antibodies can either be non-covalently bound to immunoadsorbents like protein A or protein G magnetic beads, or covalently coupled to a solid phase matrix. There are several types of immunoprecipitation techniques to know about. First, we have individual protein immunoprecipitation, IP. This involves using an antibody specific to a target protein to isolate it from a mixture found in sample solutions like crude lysates from plants, animals, or body fluids. Next is protein complex immunoprecipitation, co-IP. This technique is used to pull intact protein complexes out of solution, allowing for further identification of the unknown members within that complex. Then there's chromatin immunoprecipitation, CHIP. CHIP lets us locate DNA binding sites on the genome for a particular protein, helping us understand protein-DNA interactions within the cell nucleus. Finally, we have RNP immunoprecipitation, RIP. Similar to CHIP, RIP targets ribonucleoproteins, RNPs, and involves lysing live cells, applying an antibody, and then extracting and sequencing the associated RNA. Immunoprecipitation consists of three main stages: cell lysis to solubilize antigens, antibody immobilization to bind specific antibodies to magnetic beads, and antigen capture where the solubilized antigen is isolated on these beads. Now, why use magnetic beads for IP? They offer high capacity and yield, with all antibodies being accessible to antigens, reducing loss during washing steps. They also provide high reproducibility and purity, as interactions occur on their smooth surface, allowing buffers to be removed easily. Plus, they're fast and user-friendly, speeding up the process and making automation possible. At CD Bioparticles, we offer a range of primary antibodies covalently conjugated to magnetic beads, ideal for immunoprecipitating transgenically expressed proteins with tags like FLAG, HA, or MIC. We also have unconjugated magnetic beads for control experiments or pre-clearing cell lysates. That's a wrap on immunoprecipitation techniques and tips. Hope this helps you in your research endeavors.